Hello, everyone. My name is Neha Kamat, and I am an assistant professor in the Biomedical Engineering Department at Northwestern University. So I'm very pleased today to share a little bit about what my lab has been doing um, and to talk a little bit about how I got here. A special thanks to Dr. Ravi Radhakrishnan for the invitation to speak at Biophysical Week. So a major source of inspiration for my group has been the structure that you see here, the cell membrane. This is a structure that's found in every single living organism. It facilitates a variety of processes from sensing to transport to protein folding and cell-to-cell communication. And what's really interesting about it is it has a relatively simple structure. It's assembled from lipids that form this bilayer you see schematically drawn here. And together with membrane proteins, this structure can drive a lot of these processes you see here, from changing shape to molecular transport to protein folding, for example. And so what we are really interested in doing in my lab is taking the structure and building it from the ground up, and then taking membranes that we build and asking questions like, how can we design a membrane-based device that is therapeutically beneficial? Or how can we design a membrane that allows us to ask a biological question about the role of the membrane in a new way? So when we build membranes in our laboratory, we work with a variety of amphiphils. These molecules can be ones you find naturally exist in cells, like fatty acids and lipids, but they can also be molecules that you would not normally find in a cell membrane. Amphiphils form from peptides, proteins, dendrimers, or even completely synthetic diblockopolymers. Each of these types of molecules has a different type of property, and what we'll do in our lab is mix and match the amphiphils that we're interested in to create a membrane with either distinct chemical or physical properties. And then we'll ask a question about how that specific change in property affects a process we're interested in, like the activity of a membrane protein or the ability of a protein that's embedded in this membrane to trigger a cellular response. So we have three major thrusts in my group, all of which surround the membrane, and all of which really involve us starting with building a membrane. So our first thrust is the design of artificial cells, these structures that mimic some of the intelligent responsive behaviors of cells. Our second thrust involves designing lipid-based materials for therapeutic applications like drug delivery and biosensing. And our third thrust involves building membranes where we can study a biological process. So here we're really interested in asking the question, why do membrane lipids change as you move across different organelles and cell types? And what is the biophysical role of those lipids in driving processes of interest? So let me walk through each of these with you. The first thing I want to tell you about is our work in artificial cells. So this is a growing area of interest that many groups across the world are really starting to tackle. And where we really see artificial cells is lying at this intersection between whole living cells as well as cell-free systems. What we've seen over the past 2 decades or so has been the design of living cells where we can go in, re-engineer their genetic machinery, and program them to do useful things like sense a molecule or produce a therapeutic molecule. Cells, though, do have their own metabolic agenda, which makes it hard to sometimes execute the program you want. It's also difficult to load synthetic molecules sometimes, as well as molecules that might be toxic to a living cell. So on the other side, we have cell-free systems. These have been around since around the 1960s, and they derive from cracking open a cell, taking the guts of a cell, and what you can do is really recapitulate a lot of the processes a cell would perform, from sensing to synthesis, but you don't really need a living cell. You just need its parts. So cell-free systems are so powerful because you can add different molecules that you want to it, things that might be toxic to a living cell, or things that are not natural. You can also embed them in things like paper to design different types of assays. However, on the cell-free side, there have been limitations. You can't inject a cell-free system into the vasculature. It can also be sensitive to degradative enzymes that can chew parts of it up and render it less useful. So here what we see is that our vision is that artificial cells can bring in some of the critical functions of whole cells by bringing those membrane features back to cell-free systems. Features that involve containment, protection, as well as some of the properties and capabilities of membrane proteins to enhance what we can do with cell-free systems. Overall, we see a variety of applications in this space, from biosensing to drug delivery to bioreactors. So let's walk through some of the examples that we've been interested in pursuing. On the artificial cell side, one of the real, I would say, areas we see these types of materials being useful is in biosensing. So there are a lot of environments, particularly water-rich environments, like our blood or groundwater or areas near soil, where you might want to detect an analyte of interest or a specific chemical change. Maybe it's a low pH near a tumor, or maybe it's the buildup of lead on the floor of a waterbed. What we envision artificial cells as being used is as sentinel sensors, particles that can move through water-rich, complex environments and carry within them some sort of genetic code, a genetic code that says, if you see this molecule, produce this protein or produce this other molecule. So that when we collect these particles downstream or in a blood draw, we can look at them and we can see where they've been and what they've seen on their travels. And so what we're hoping and what we envision is that materials like this will really advance the types of information we can get out of water-rich environments. So this is an example of a type of vesicle-based biosensor that we've been working with that we've recently archived as a preprint. Here we worked with our collaborators in Julius Lux here at Northwestern, and we encapsulated an RNA riboswitch that they work with in their lab, as well as a cell-free extract. This is a fluoride riboswitch, so it's responsive to fluoride. And in response to interacting with fluoride, this riboswitch will become active, allow for transcription of a gene, and subsequent translation of any protein that's encoded along this gene. And so using this, we designed a sensor that could sense fluoride and in response, produce GFP, a fluorescent protein. What we showed in this study is that the membrane provides a lot of critical features. It protects the cell-free extract from degradated enzymes, while still allowing our analyte of interest to cross the membrane. It retains all of the molecules that we need to keep our cell-free system running. And by changing carefully the composition of our membrane, we can change its permeability, allowing less fluoride or more fluoride to enter our vesicle sensor, which has a direct effect on the sensitivity and performance of the total sensor. This is an example of work we're doing in our second thrust, designing membrane-based materials to control cellular behaviors. Here, we used a protein called TRAIL, or TNF-related apoptosis-inducing ligand. TRAIL is a ligand that has been extensively studied as a target for cancer therapies. It can selectively trigger cell death in tumor cells, and it's a homotrimer that can bind to 5 different types of receptors in a target cancer cell. DR5 and DR4 will effectively trigger intracellular apoptosis, but DR2, 1, and this OPG receptor are decoy receptors that bind TRAIL and do nothing. One of the things that we learned about these receptors is that for DR5, specifically, it needs to be clustered and organized in the cell membrane to start to initiate that apoptotic cascade. And so we wondered, could we use design features in our lipid vesicle to control the presentation of TRAIL and better trigger apoptosis in the target cell? So here what we've been doing is playing with this idea of phase segregation. Lipids of different composition and different physical properties can phase segregate into specific regions. We know this happens in a cell membrane, and it also happens readily in lipid synthetic vesicles as well. What we did was we conjugated TRAIL to a lipid through a histidine nickel conjugation mode. And what we showed is that we could control the spatial arrangement of TRAIL so that it was either uniformly distributed across a nanoparticle, or if we could phase segregate the lipid that it was attached to, we could also phase segregate the TRAIL itself. As a result, what we found is that the spatial arrangement of TRAIL has a direct effect on its potency or its ability to induce killing in a target cancer cell. Briefly, what we found is that if we clustered TRAIL into these lipid domains shown here, what we could do is we could better pull together that receptor DR5 and more efficiently trigger apoptosis. So we have been really excited about this idea because what it shows is that we can use phase segregation in lipid nanoparticles to control the spatial arrangement of target proteins. And what we've also shown is that the actual spatial arrangement of proteins on your therapeutic nanoparticle is critical in terms of how a cell might respond. The final thrust I want to discuss with you is our membrane mechanobiology thrust. And so here we've been inspired by this idea, or really, I suppose, curious about this phenomenon, where different cell types and different cell organelles have very different lipid compositions. And we wondered why. Why do different types of cells and different types of organelles have different compositions? Specifically, one of the questions we have is, when these cell lipids change, what is the effect on the physical properties of the membranes they assemble into? What's the effect on the elasticity, its ability to stretch, its bending rigidity, its ability to move out of plane, the permeability of a membrane, the thickness of a membrane, the fluidity of a membrane? And how do these properties impact a variety of protein-based behaviors, from folding to activity to the location of proteins? Can we take what we learned with our model systems and thereby allow us to understand how cells use lipid changes to regulate their functions that involve membranes? So an example of this is we've been really curious about this phenomenon called protein folding, which we often think about in terms of soluble proteins, but there are a variety of membrane proteins that need to be folded properly in order for a cell to function. And one of the questions we've been asking is, how do the properties of membranes actually impact how efficiently a protein folds? So to answer that question, we've been using cell-free systems where we will take all the components we need to make a protein, along with a plasmid that encodes the protein itself, and we'll throw in vesicles of different compositions. What will happen is, as the protein is being made, encoded by these genes here, it will co-translationally insert into the vesicles that are present. And we use a reporter assay shown here, where C-terminal GFP protein tells us how well the protein folded, and as well as gives us kind of a readout for how much protein was made. With the system, we've been finding some really interesting things, such as the membrane properties really affect co-translational folding and synthesis of a variety of membrane proteins. And what that leads us to conclude is that one of the roles that lipids have in a membrane is really to facilitate folding, specifically in the ER, where a lot of that folding takes place. So this is a question that we have been digging into in more detail, but one where cell-free systems together with synthetic vesicles have allowed us to probe this relationship in a very new way. So to wrap this up, I briefly want to tell you a little bit about how I got here. This is a timeline of my research path. I was born in South Bend, Indiana. My first experience with research really came about, or a significant experience with research came about when I was in high school and had a really terrific science research program. This was a public high school. It was a new concept at that time to have science research programs, and I was really fortunate to have access to it. Through this program, we got partnered with different labs at our local university, which was Notre Dame. And there, I started working with Professor Joan Bronicky in their chemical engineering department, looking at the toxicity of a substance called an ionic liquid. And I really sort of started to fall in love with research and the fact that no one really knows anything when you start out, and it's up to you to sort of create the narrative by finding out what is actually going on. And I really enjoyed that independence and freedom and really the unknown of it all. From there, I was fortunate to head to Rice University, and because of my previous research experience, I had the opportunity through a scholarship called the Century Scholars Program to start research right away. So I started working with a professor called Jennifer West, working with gold nanoparticles, and I continued working in this lab all 4 years, and through, I think, all of my summers in undergrad. And in this program, I really cemented, I think, I fell in love with research all the way. And Dr. West was a wonderful mentor, who is now at Duke, but who I continue to stay in touch with. And so during this time, I got to be part of what's called the Beckman Scholar Program, which is a 2-year program where you get to pursue an independent research project that you have designed yourself. And so I really enjoyed that experience. I did during that time try clinical internship and took the MCATs to see if med school was for me, since all of my friends were going that route, and also went on some consulting interviews just to see what was out there. I really discovered that research was my passion, and so I continued on to graduate school at UPenn in their bioengineering program, where I stayed for 4 years working with my advisor, Dr. Daniel Hammer. And so here is where I really began really fell in love with membranes. I worked with polymer vesicles, a structure that Dan co-invented, and these are really lovely membranes that are assembled from synthetic diblock copolymers. And so here I also became interested in this idea of building artificial cells. So I realized that membranes were the scaffold for a cell. And wouldn't it be great if we tied in that other critical component of a cell, its genetic machinery? And so I knew I needed to learn about the genetic machinery of a cell. And so for that, I went to pursue a postdoc with Jack Shostak at Harvard and MGH. Jack won a Nobel Prize in 2009 for his work discovering telomeres, and his lab has been investigating the origins of life and specifically looking at the role that RNA can play in sort of under prebiotic conditions to replicate, and how that replication can happen within cell-like structures. So I had a really wonderful time working there for 4 years, and following that, was able and was very fortunate to get a position here at Northwestern in the biomedical engineering department, where I've been sort of bridging my molecular biology knowledge as well as engineering knowledge to design artificial cells and study membrane mechanobiology. In my last minute, I just want to say there are quite a few people that made this path possible, and so I just want to acknowledge them. From my high school teacher, Mr. Nevin Longnecker, who is still teaching currently, to Joan Brennicky, who I did my first high school research with, Jennifer West, who was at Duke University. Actually, she's now a dean at UVA, Professor Daniel Hammer, who really encouraged me to pursue academia at a time where I needed that encouragement, and will always be grateful. And Jack, who's taught me a lot about how to get really involved in your science, no matter, I was always inspired that no matter how big his lab was or how busy he was really intimately involved in every project in his lab. And then, of course, I think the people who you really, really work with when you join the lab, the graduate student mentors. This is Amanda Lowry, who I worked with as an undergrad, and Greg Robbins, I'm not sure what he's doing right now, but who I worked with as a graduate student. And they were really instrumental in me getting a good start. Last person I want to thank is my dad, who started doing science fair projects with me when I was in middle school, and who introduced me to a laboratory setting when I was really young. And so I just thought it was normal to be in the lab. And I think, wouldn't it be great if we all had that kind of access to science? And wouldn't it be great if we had more people in science that looked like us? So with all these mentors in mind, I want to thank them and thank you for your time and your attention. This is my wonderful lab. I'll end here. They are a terrific group of people, and this job has been really fun, and I look forward to spending hopefully many more years getting to do this. So thank you for your time.